Welcome to the latest edition of the Mind-Gut Conversation podcast, a place to learn about breakthroughs in the science and practice of health, mind-body interactions, the microbiome, food, and the environment. Today, I have the great pleasure to talk to Dr. Tracy Bale, who is the Anschutz Foundation Endowed Chair in Women's Integrated Mental and Physical Health Research in the School of Medicine at the University of Colorado, Anschutz Medical Campus. Dr. Bale is also Professor and Director for Intergenerational Stress in Health and the Director for Sex Differences Research in the Department of Psychiatry. Dr. Bale's research focuses on understanding the role of stress dysregulation in neurodevelopmental and neuropsychiatric diseases and the sex differences that underlie disease vulnerability in humans. She's interested in stress and adversity across the lifespan, including examining the effects at the germ cell level and the mechanisms involved in altering brain development. Dr. Bale's lab examines these mechanisms in preclinical models, attempting to translate research findings to humans to identify those processes and biomarkers important for promoting disease risk and resilience, especially in vulnerable populations. Dr. Bale serves on many international and internal and external advisory committees, panels, boards, including the NIMH Board of Scientific Counselors, and has been the recipient of numerous awards for research, including the Richard Weitzman Memorial Award from the Endocrine Society, the Medtronic Award from the Society for Women's Health Research, and the Daniel Efron Award from the American College of Neuropsychopharmacology. She is the president of the International Brain Research Organization, IBRO, and was awarded Top 100 Women in Maryland 2020. Welcome to the show, Tracy. I'm really looking forward to this conversation, talking to you about your work and your interests. So let's start with a general question about the multiple ways by which stressful environments that a person experiences, either preconception, during pregnancy, during early development, influences the phenotype of the offspring. And so phenotype meaning behavior or health or disease. Could you discuss some of the most important aspects of this intergenerational programming and some of these examples, main examples, programming of the HPA axis, the vaginal microbiome, maternal gut microbial metabolites, and inflammatory signals affecting the brain development of the fetus, and extracellular vesicles, something that you have in your research focused on. And whatever you want to emphasize of those mechanisms, we can focus on those.
Sure, thanks, Simran. I'm going to start from the first thing you mentioned, which is programming of the HPA axis, which is the hypothalamic pituitary adrenal stress axis. One of the reasons that we've spent all of my career basically focused on how your stress response matures, develops, and is programmed by your experience is because it is the most translational part of the physiological response that we all have in terms of how the hormones that are produced, the system in which it feeds back and shuts back down for anything from an acute stress experience. Again, if you're driving on the freeway in LA full of traffic and out of nowhere, you're daydreaming, someone pulls in front of you and you have to slam on the brakes to avoid an accident, or you get into an accident, even worse. That's what we think of that as an acute response. And you will have a hypothalamic activation producing hormones from your pituitary that will activate your adrenal gland, making those stress hormones we know as glucocorticoids. One of the reasons that my whole career is spent using that as a readout is because it's the same in all mammalian species. And so unlike things like trying to understand mood in a human being and trying to model that in a mouse, which is very difficult. How do you know what the mood of a mouse is? Or many of the behavioral tests that we often use in neuroscience. These are very complicated to sort through, and without being able to have a conversation with the species, you can talk with a human being, how have you been feeling? Can you get out of bed? How has your sleep been, et cetera? Many of these things are very difficult to assess in a rodent. So the stress axis is very easy because you can take a very small blood sample and have a pretty good idea of the state of that animal. So that's an acute response. We can also learn a lot about chronic experience. So just like in humans, where you can experience chronic stress, maybe your job is very stressful, maybe your home life is very stressful. You can also experience kinds of trauma. Those could be sexual trauma, kinds of interpersonal violence. You can experience, for instance, in this country, if you belong to a lot of minority populations and communities, you may experience different kinds of trauma, discrimination, microaggressions, bias, et cetera, lack of equity, lack of resource assessment. And those kinds of traumas can be very impactful in current generations and in future generations as well. And I think that understanding chronicity in our animal models and even in the level of cell culture allows us to assess a lot of mechanisms that can then be translated into therapeutic interventions. And so that's the HPA axis. And we know that those glucocorticoids, the stress hormones produced in all mammalian species, they not only serve to tell the brain, but also the body that this is stress. And I think one of the really important things in this field that we think about is that all mammals have a circadian rhythm. And while some species are nocturnal and some of them are diurnal, that those glucocorticoids are actually part of that circadian rhythm. And so every cell in your body sees stress levels of glucocorticoids every day in a 24-hour cycle, which is super interesting. So how does a cell know that you've experienced stress? The example I gave you, almost getting into a car crash on the freeway, versus the rise and fall every day of rhythm. And that's one of the molecular or the mechanisms within individual cells. You can look inside a neuron. You can look inside cells that line the reproductive tract. And those are the cells that we're also interested in my lab, because if you change the programming of a cell along the reproductive tract of mom or dad, now you have the possibility of affecting which signals go into the germ cells that become the next generation. And so while germ cells are constantly turning over, especially in men, right, you're making new sperm every day, that in fact, the glucocorticoids, those stress hormones under trauma or chronicity of stress, can actually change those cells along the reproductive tract, changing signals that tell the germ cells, this has been a stressful environment. We need to either kick up the rate of fertility or decrease the rate of fertility. And you can imagine, Emran, that during particularly traumatic events, such as a famine, right? Evolutionarily, what's the greatest disadvantage for propagation of a species, survival of a species? Famine. So during that, you may not want to reproduce. But when there is a signal that that traumatic event or stress is over, now you may want to quickly propagate a species. And so we can say that. We can see that in a cell culture system. We can see that in the mice that we use. And we also have human studies that we follow along in many different traumatic or adverse events.
So this happens on a this happens on a biological level, not necessarily on a cognitive or
exactly at a biological level, very much so. I mean, of course, in humans and in animals, you need to have the perception of stress itself. So for mice, that can be something that we expose them to every day for weeks on end that makes them very stressed. They don't like change, they don't like novelty, and that tends to produce lots of stress hormone for them. For humans, we study cohorts at which different communities have been exposed to, again, discrimination or bias or interpersonal violence, such as sexual trauma, or maybe things such as different periods across the lifespan, Emran, really matter for when trauma happens. So again, you're surrounded by a community of a lot of gun violence. That might be something that as a child becomes part of how different physiological systems can mature and can ultimately change how you might produce offspring in the next generation.
Yeah, this is really fascinating. As a side question, I mean, there's this phenomenon of decreasing fertility in quite a few Western countries, extreme in some countries. Do you think this is in any way related to this?
That's a great question. I've actually been on several different panels recently around the world talking about some of the effects of the current escalation in stress around the world that we're all experiencing. Climate change has become incredibly stressful depending on where you live. As Emeren, as you and I were just discussing, the floods, fires, landslides, all of those things that are happening in California are all a big part of climate change, and they're not going away. And if you have a house, depending on where you live or the traffic you have to deal with, et cetera, it's a continuous stressor that you endure. In other parts of the world, flooding, if you live on an island, et cetera, crop production, food availability, all of those things. So that's just climate change. Now, if you add on top of that, the political dysfunction that's happening in many countries, including our own, you don't have to even pay attention much to the news. All you have to be on social media just to see the divisiveness. And then you have the pandemic. And especially for our young people, we're seeing a huge escalation, 30-40% increase in mental health disorders. And so all of these things together, yes, they're producing an incredibly stressful environment, much of which is out of our control. And we know that that's the worst kind of stress, that which we have no control over. And so is that contributing to the reductions in fertility rates? Possibly. Could it also be a lot of environmental toxicant factors that they think might be playing a role as well? And there may be some feedback evolutionarily about populations, control within populations in cities, et cetera, that is evolutionarily trying to control itself through fertility. So that's the HPA kind of. I think of that as the higher order aspect of the studies that we do, and of course everything mechanistically trickles down from there. Whether it's Emeren, you mentioned the maternal microbiome, whether it's maternal vaginal microbiomes, and yes, your vagina has a microbiome which is unique to each individual woman, and your gut, of course, a lot about out there, and thinking about the gut microbiome. So all of those play roles in the diet that mom has access to. Again, you can think about our vulnerable communities who don't necessarily have access due to food deserts and a lack of stability and resource access, communities where they may or may not have resources available for producing or consuming nutritious food, and therefore the gut microbiome in a way is very affected by that, and the metabolites and nutrients produced and processed by the microbiome differ, and that can affect mom's health, which affects baby's health, or even more directly have an impact on fetal development. When you're born and you're a natural vaginal delivery, mom's vaginal microbiota environment can be very impactful for everything from the baby's skin microbiome to that bit of inoculant that babies get, because going through the birth canal is a little bit like passing through a very tight vacuum. And so there is ingestion of some of that vaginal microbiome. Our group and others have identified the fact that initial inoculant, we like to call it, that initial seeding of the baby's otherwise empty gut microbiome can be really important for programming initially the immune system. And now we set off all kinds of possibilities for how, from your work and books that I'm looking on the back of your shelf there, Emeren, the importance of the gut immune connection starts, from the point of birth. And so those are important mechanisms. And all of that, of course, feeds into how the brain develops, because as a neuroscientist, I care a lot about the brain. And then the last one, the extracellular vesicles, is a much newer mechanistic biomarker and signal that we've been studying in the lab for probably about a decade now. A lot of these studies came out of the cancer biology world. Cancer biologists have been studying vesicles. These things are these nanoparticles that are secreted. Again, I love the translational piece of this, because all mammals produce them. All tissues in your body produce them, and they travel in circulation at very high concentration, but they have an incredible specificity, despite the fact that they're so dynamic. They have very specific proteins and very specific types of these vesicles, these nanoparticles that have a mission. They're out to do something. They're a signal for how, for instance, in pregnancy, how the placenta, which produces a ton of these vesicles. So in the stage of pregnancy, there is a more than 4-fold increase in concentration of circulating vesicles. And why? Because they're doing all kinds of crazy important things. The placenta secretes them to coordinate and strategize the cells across the placenta based on maternal health and programming of fetal health. And so they're really important communicators. They also coordinate for mom. They travel in mom circulation. They do everything from coordination of the circadian rhythm and coordination of glucose uptake. So if you're pre-diabetic, you might be producing more of these vesicles as the body tries to compensate for getting glucose taken up faster. So they're super fascinating, Emran. We've been able to do studies in human cohort populations for different communities exposed to different types of trauma at different time points in the lifespan. So taking, for instance, 30 and 40-year-old women in the city of Atlanta that are working through the Grady Trauma Hospital and actually looking at a traumatic experience that they had decades earlier and being able to isolate from blood samples specific proteins that have led to a whole new field of study about how integration of the autonomic nervous system that innervates the skin, which is something we had never thought that we'd be studying. So they seem to be stress signals, they seem to be stress responsive, and they may serve as really important biomarkers that could tell us, for instance, a pregnant woman who might be at risk of any number of insults in pregnancy, that she could be better watched and intervention to prevent morbidity and mortality for both mom and baby.
So are these vesicles, I mean, it's obviously a way that the body and cells and organs in the body communicate with each other, but is there evidence that they actually play a role in disease, like alterations? Like you said, there's this massive increase in placental.
Yeah, there is some evidence out there that you see changes or composition, there's changes in size, there's all different things you can look at for these extracellular vesicles. In cancer, for sure, they've done a ton of work. There's startup companies and patents everywhere around using these as both biomarkers of disease state, but also in producing synthetic nanoparticles that either could have a direct targeting where you could utilize them to deliver drug targets. People are now looking at them because they come in and out of the brain as well. Is this a way to get beyond the blood-brain barrier? Could you deliver drug delivery to really specific targets to avoid side effects, for instance? It's becoming more and more. I presented on this about a decade ago at the big neuropsychiatric conference, ACMP, and it was the first mention of these things at the meeting, and people are like, what is this? This looks like magic. And 10 years later now, it is actually something that a lot of research in psychiatry is looking at is this a way that we can say what treatment might work better for somebody in psychiatry, as well as a biomarker, meaning of state of an individual, right? We don't have any biology. If you're depressed or anxious or schizophrenic or PTSD or whatever you're trying to have treated, there's no way other than your feedback of how I feel to say biologically the drug is not working. And so this might be one of those ways to say, hey, we're seeing a reduction in a particular biology of these markers.
I mean, I can't help asking the question, a microbiome related question. Is there any communication between these
good
vesicles?
Question, Emran. There actually is. So bacteria produce their own nanoparticles. So the name has changed over the years as people have begun studying them more and more. So extracellular vesicle or exosome is something that mammalian cells produce. Different bacteria, depending on the cell wall composition of the bacteria, make their own type of these same nanoparticles that we know communicate across the gut and this is where they've been studied the most, across the microbiota, but also now seem to be maybe detectable in circulation. So could they be signaling to other organs or cell systems within the body as well? it's a brand new area. Not a lot is known, but yes, you definitely see these same particles produced by bacteria.
It's fascinating because, as you know, there's also now this interest, intense interest in the role of the gut microbiome in cancer treatments and in melanoma, seemingly predicting the outcomes. Depending on what kind of composition of microbes you have either a positive or negative outcome from the conventional therapy. I just wonder, just listening to this conversation, if these exosomes are not involved in that phenomena, because
the longer I've been studying them, Emma, I would say it seems like they're involved in just about everything. There's now evidence within the brain that these things serve as local communicators across synapses and participate in how glia may communicate more specifically with neurons. So I think this is going to be, a coming decade of really trying to understand. 20 years ago, people thought these were garbage. They thought that the cells were dispensing of all the cellular garbage, and that's what they were traveling in circulation. It's really only been the last decade or so, again, a lot done by cancer biologists to figure out that there's such cell communicators.
Yeah, so really fascinating to listen to you, I have to say, so let me ask you this question. So from the microbiome world, I'm most familiar with, we're already dealing with hundreds of thousands of metabolites that are being generated, and that can, many of which are getting to the systemic circulation. And so now you have all these other signaling molecules, including these exosomes. How does the organism deal with that massive amount of information that's receiving?
I don't know, Emma. That's a great question. I mean, when I first started working in extracellular vesicles, which came through one of our findings related to doing omics, transcriptomics, looking at broadly across changes in the placenta in mouse and human, and sex differences and stress and what was happening. The regulatory mechanisms that we kept coming back to again and again seemed to regulate these proteins. And the more we looked into what these proteins were and how they were being regulated, it was part of this mechanism of secreting vesicles. And that's how we got started in this area. And then learning more about what they do and how do they, signaling locally seems obvious, so intergenerational studies that we do in males have a lot to do with how sperm matures. And when you go through spermatogenesis in the testes, when sperm are at the end of that spermatogenic cycle, they're not mature. They can't swim, they can't fertilize. And so it's kind of a fluid momentum that pushes them into the epididymis, which sits on top of the testes. And there's actually a host of activity that has to happen in that epididymis, and it's called a caput, which is this very specific part of the epididymis. And so it's not just storage, there's actually active maturing of the germ cell that happens there. And because now it's outside of the protected blood, I want to say blood-brain, but blood-testis barrier, and there's all kind of jokes that we'll leave out of that aspect of the similarities between the blood testis and the blood brain. But in the epididymis, it's now that dad's DNA is protected, right? Because remember, it's all during the process of spermatogenesis. The point of doing that is that in addition to replication, the DNA is all wound up very tightly and protected in these protamine structures. And so now that you've safeguarded the DNA from the environment, now, if there was anything evolutionarily that made sense that could be beneficial to your offspring, do you want them to metabolize energy better? Do you want them to conserve energy? Do you want them to be born faster? Whatever. That's where you'd want that environmental signal to happen. So we spend a lot of time in those cells that surround that function. And so one of the things that's critical for that maturation point are extracellular vesicles. So that makes sense. Like, when you're talking about like, how does the system regulate all of these signals in the systemic milieu? Like, well, locally in the caput of the epididymis and these tubules there, that can make sense, right? The EVs, that are circulating there, talking to the sperm, and it's local. So to me, like, okay, that makes sense. You can change what the cells are putting into those vesicles, evolutionarily conserve that, and that could change as the sperm deliver those signals to the egg. You could change something dramatically about the offspring. But you're right. When you start thinking about, okay, now things that are secreted into circulation coming from the placenta in pregnancy or whatever, right? 4 times the concentration. Yeah. How does a system deal with all of that communication? Well, I will tell you, I think it's similar for a lot of microbiome metabolites as it is for EVs. They're really dynamic. They're turning over very quickly. And so I think not every signal is going to make it where it needs to go before it's washed out of the organism. And maybe that's the limiting factor. And that's why in pregnancy, where it's so important, you increase the concentration, because now just by the law of averages, you're more likely to get something where it needs to go.
Interesting.
Yeah. So maybe it's just the fact of everything's going through, some things are going to be taken up, some things are going to wash out. And if it's really important, that concentration is just going to be higher.
Yeah. So let me come back. So was the first question, we talked about these multiple ways by which a stressful environment is transduced, either for people that live in that environment or for the offspring intergenerationally. But not everybody gets sick, or not everybody develops a psychiatric disease. So there must be some very powerful counter-regulatory resilience mechanisms.
Yeah.
Do we know anything about those?
Yeah. So I think about this question a lot. The kind of the old terms we used to use are risk and resilience. We try to stay away from resilience a little bit, only because if you're out in the community and you're framing it as risk and resilience, saying the term resilience places the onus on the individual, is that, well, you must not be tough enough because you can't overcome this trauma. But in science, we think of it as risk and resilience. And I think what you're getting at, Emran, is why even within a given household experiencing, for instance, the same trauma, dad is abusive or mom is neglectful or what have you, how is it that different individuals rise out of that? Or even actually, there's terms in psychiatry where they utilize that as a launching point, where some individuals take adversity and use it as a way to make themselves stronger, and some individuals can't. And I think of this as what I term as the G by E by D, which is your genetic background that you inherit and you can do nothing about. And even within the same family, you may get different risk and resilient SNPs, so small nucleotide polymorphisms, that you may have the combination because of, and I will say that sex and gender play a role here, very much so. And so you may have a composite of DNA that just puts you at risk, and no matter how much you would like to overcome your adversity, you don't have the tools or you're necessarily the DNA to do that. So that's the G is the genetics, the E is the environment that you're talking about. And so it also may be part of that, right? So maybe the household is full of adversity, but one of the children happens to have a friend whose household where the parents are actually nurturing and they spend a lot of time over there, or there's somebody at school that takes them under their wing, whatever. So that's the E. And then the D is the developmental windows, because it does matter when adversity happens. Did that adversity happen preconception, during pregnancy, early in childhood? We have some of these biomarkers, these EVs, have shown us in humans that with particular types of traumatic events, we looked at sexual trauma because you can really identify the timing of when it happened. And it produces such remarkable differences in health risk. So if that adversity happened in childhood, way prior to puberty, those women were really at risk as a group, clearly, for obesity, diabetes, and hypertension, which is probably a composite of programming that happened. Probably involves something about the immune system, may involve the microbiome got set up, right? How you process energy. if you experience that adversity during that adolescent window, prior to adulthood, but during that window, you didn't show any of those effects related to obesity and diabetes, but man, you had a cascade of risk factors for PTSD and holding on to the trauma, even though for both groups, it was decades later. And again, we saw these differences with the biomarkers. So I think it's the complexity of who we are as human beings with the DNA we inherit, the environment we experience, and when both adversity and enrichment, right? The other side of that coin, when it happens, if you experienced a traumatic event, but you had a really nurturing environment at the right time, it may wash out those effects. And sex as a biological variable, man, it is so important. Boys seem to be much more at that risk point prenatally, all kinds of adversity that happens. And we've done these studies in mice and we've studied them in humans. And, you know, if you ask a NICU nurse or an obstetrician, they will always tell you there's more boys than girls in the NICU, that if there are girls in the NICU, they go home earlier than boys. There's just something protective. We think it has a lot to do with the programming of the placenta that seems to protect girls in utero. But once you're born, that flips around. And now a lot about the sex differences in DNA puts girls at greater risk. So I think it's a really important but very complicated question that involves a lot of those factors that we're working on studying. But I think we can't really dismiss the idea that boys and girls are, again, different windows of vulnerability.
It's really fascinating because, as you know, like one neurodevelopmental disorder, autism spectrum disorders, is more common in boys, and
actually, Emern, all neurodevelopmental disorders are more common in boys than girls, which points to that in utero.
Yeah.
Yeah. No, it's
interesting, you know, as you know, like with the microbiome science, this has been very, a lot of people have jumped on this in terms of autism spectrum because the patients with that diagnosis have an altered gut microbiome, but
they also have an altered diet, which probably contributes to their altered microbiome.
Yeah. But what I always thought is it's much more interesting and important, given this developmental trajectory, what happened to the pregnant mother, you know, because obviously this autistic brain did not develop after delivery.
Nope.
It developed in utero, and
exactly right.
Yeah.
And so the big question then is, you know, you will get some people who argue for autism diagnosis that girls hide it better than boys, and so they're diagnosed less. But I think if you look at the data just overall, even in populations where, you know, is it also access to diagnosis, who's likely to go in, whose teachers are likely to report disruptions, those kinds of things, that it really still holds up, right? That there's more boys than girls. And so then if it happens developmentally, do our sex chromosomes contribute to the risk and resilience? We think so. There's some really protective mechanisms that escape exon activation. So there's more of them in the cells of a girl's placenta than a boy's. And how do those factors factor in for all of the things happening? I mean, pregnancy, it's a huge insult to mom, right? Physiologically, it's a big demand she's carrying. So how does that whole system regulate? Like you said, mom's gut microbiome, mom's vaginal microbiome, all of those things factor in.
And that's only recently that people have focused their attention on it, certainly in autism. And I think the same thing will probably hold true for schizophrenia, and
absolutely
potentially also in the long term, you know, for these neurodegenerative disorders that they may have early.
Right. There's new thinking that neurodegenerative disorders are actually neurodevelopmental in origin, which makes the year span in humans very difficult to figure out the linkage, right, of what you were exposed to. But probably very important.
Just mention something in your CV. So in your current position, you also do a lot of activities with community outreach and education, and maybe you want to explain some of this. I found that fascinating. Of all the people that I've talked to on the podcast so far. I've not seen that in their CVs. So
Yeah, I think it's incredibly important. The thing that we started off this conversation with, Emran, about the divisiveness in this country right now, just as Americans. I'm also president of the International Brain Research Organization, which has somewhere close to 80,000 members around the world. We have 5 regions that literally in neuroscience encompass the entire world of neuroscience. And so you see where equity is lacking and where diversity is not valued and bias exists. And I think even within the United States of America, because of the incredible divisiveness that is not going away anytime soon, that if we as scientists don't engage in our communities, and I am not talking about outreach, and I'm using air quotes here because I really don't like that term. Outreach suggests that we are somehow privileged out doing things for individuals in the community, but rather engagement, which says, we're going to learn from you and you're going to learn from us, and we're going to have a conversation to benefit each other. If we don't do that, then we miss the opportunity to step out of our ivory towers as we're perceived here, and recognize that the things that we write in our grants and the things that we study are actually currently existing and happening in the community. And I study stress and trauma, and there's a lot of that out there right now. And understanding what a community perceives as their concerns, what a community perceives as its needs, what a community decides that they need help with, is something that we can't determine for them, A. And B, when we talk to our communities and engage with them, they learn about the science that we're doing. And nothing could be more important right now than retaining the fact that science is science and factual, rather than this perception that it's an opinion, which is our own doing, because we're not out in the community and we're seen as being the elite doing these things that we don't care about them being the community. So I think that it is incredibly important. And so in Baltimore, I spent 5 years developing programs to really invest in a conversation with different community members, different community organizations, schools. And it's amazing how programs just grow. And so we did school-based programs of using science to talk about stress and trauma, to use reading as a positive way to deal with stress and trauma. And from that, doing art. We have a whole art program that we also did around the science, that then brought in the mayor's office, and the mayor's office invited me to work with city council, and then city council invited me. And then from that, which was incredibly rewarding, Emran, began a conversation of city council members working on trauma-informed care for policy that affect the laws and governance of a city and a state, to then say, hey, you know what, there's some biology here. Let's talk about the biology of trauma, which then became instituted into the laws and the policies in a way that gave perspective to how you might think about developing laws, and then fed into not just governance, but fed into policing, which in many communities is a huge issue right now of how policing happens. And so it was incredibly rewarding. I learned a lot. It was rewarding working with those community organizations, people so passionate and work so hard to ensure equity within a community and access to resources, right? You can have things available, but if you didn't build your city in a way that the communities that need those resources can access them, then it's useless. And so it was, we had city council come to campus and they did research in the lab. And it was really rewarding. And in the end, we built this huge trauma summit where I've invited scientists who study trauma to educate city leadership and community member leaders, and then vice versa. Here's what we're seeing in the community. Here's the power of what's happening amongst the different community members and the policies and equity that's happening to inform the scientists. So it was a huge thing to organize, I will just say, but it was very rewarding, very rewarding. We got the mayor's office and the chief of police to come together in one forum, which, if anybody out there knows Baltimore, knows that that's difficult. That's really difficult. So it was great.
Well, that's a wonderful way of ending this conversation. I don't want to end it. Hopefully, we'll have many more opportunities in the future to follow up on this.
Always happy to talk with you, Emran, and thank you for all of the incredible work that you do. You know, putting science into books that the community can digest, I think, is also an incredible value. So, thank you.
Thanks again, Tracy. Thanks.
Sure.